Hello, I'm Benjamin Miller, and today we'll be talking about designing antibodies in Rosetta. This talk is focused on the publication RosettaAntibodyDesign, a general framework for computational antibody design published in 2018 in PLOS Comp Bio out of the lab of Roland Dunbrack. Rosetta has been used extensively for design and design plus docking applications. Many papers have come out of Rosetta Labs focused on some aspect of protein design and docking. For instance, this paper from the Baker Lab in early 2021 is looking at designing proteins that bind to the transferrin receptor. However, that publication developed a bespoke protocol for that system which may not be amenable to your specific needs. RAbD, while specific to antibodies, offers a flexible, general framework with which to design antibodies for your needs. It can be used for many design projects, including affinity maturation, homologous redesign, stability improvement, and de novo design. While this workshop is antibody-focused, in case you have come across this lecture divorced from its context, allow me to take a few moments to go over the antibody, specifically the IgG, structure. IgG is Y-shaped, made up of heavy chains, colored in yellow and blue, and the shorter light chains, colored in green and pink. The antibody is further broken down by region: the Fc region, or crystallizable fragment, the Fab, or antigen binding fragment, and the Fb, or variable fragment. The Fb region is where the specificity and diversity comes from in the antibody, and is the region that we'll be focused on for the rest of this talk. The Fb region contains 6 complementary determining regions, or 6 loops, 3 on the heavy chain and 3 on the light chain. These loops are highly variable in length, sequence, and structure, and are what we'll be primarily designing. One of the main features of the algorithm is a sampling of a database of CDR loops. The Dunbrack Lab has previously published papers on the clustering of CDR loops in antibodies. The method was first described in 2011 in the Journal of Molecular Biology, and then in 2015, the database and associated web server were published in Nucleic Acids Research. The protein databank was queried for all antibodies, and any low-resolution structures were filtered out. CDR loops were separated by type, L1, L2, etc., and further by length, 10 residues, 11 residues, etc. The loops are then clustered by a dihedral angle metric, so loops with similar shapes are clustered together. In the figure, we see L1 loops from the PDB, filtered by length and the shape metric. On the bottom, we see the label for each group, L1-11-1, which means CDR loop L1, length 11 residues, and it's the largest grouping, so it gets labeled 1. The second largest group gets labeled 2, and so on. Below that is the sequence logo for the group. While I encourage you to read the paper, the PI-IG Classify server, which I will discuss on the next slide, is the best place for the most up-to-date information. This is the homepage for PI-IG Classify, a web-based server with the CDR loop database required to run RAbD. In addition to the database and a classification of all antibodies in the PDB, it also allows the user to submit an antibody of their choosing and will identify antibody chains, antibody gene, germline, CDR/framework positions. It will also renumber your structure to AHO numbering scheme and classify the CDRs. This renumbering is essential for properly using your structure in RAbD. Now that you understand about loop clustering, let's discuss the RAbD algorithm. The RAbD algorithm follows this general workflow. The outer loop randomly chooses one of the CDR loops and replaces it with a loop from the database. Then, the inner loop is entered, where the algorithm performs sequence design and local structure optimization. After each design and optimization is completed, the new model is either accepted or rejected according to the Monte Carlo criterion. This cycle repeats for n cycles. After n rounds, the inner cycle are concluded, and the final model is then compared to the previous best outer loop model, and accepted or rejected based on the Monte Carlo criterion. Typically, n outer cycles are set to 25. The algorithm is very flexible, however. This is just a general overview. What I've just described does not need to be run in its entirety, so please refer to the tutorial for all the potential options. Now you're probably wondering, does the algorithm work? In the initial RAbD paper, the authors sought to answer that question by experimentally validating their results by improving the affinity in two known antibody-antigen complexes. One antibody that binds to the main allergen in bee venom, and the other which binds to HIV GP120. Both have published crystal structures. Design was performed by swapping some, but not all of the CDR loops. Approximately 30 designs were chosen for each antibody, and 3 anti-bee venom antibodies had improved binding, see the figure on the left, and 1 anti-GP120 antibody had improved binding, the figure on the right. While a few designs improved binding, this is a successful result, given the low number of constructs produced and that expression, purification, etc. are always potential roadblocks. Now this is just a brief overview of the tutorials. The tutorials themselves are found in a different video. The tutorials are broken down into 4 main sections with multiple parts per section. The first is sequence-only design, mutating the sequence without swapping the CDR loops. The second section involves grafting new CDR loops onto the antibody in addition to sequence design. The third section is de novo design, starting with random CDRs instead of the native. And finally, section 4 gets you on your way to creating your own protocols using Rosetta scripts. This is the end of the presentation, and I hope you will check out the next video where we walk through the tutorials. Thank you.